Okay. So in a cystosomes, as you can see, the cystosomes is one of the trematodes, and remaining trematodes we are going to see today. In that trematodes, some of the first we look for generally for what are the general characteristics of these cystodes, general characteristics of these trematodes. So that you must know, because first you must know about the parasitology, that is the helminthology, what part we are now teaching you about the helminths. In helminths, what are the different parasites are there? The worm-like parasites, some of the flatworm-like parasites. So that you have to differentiate like cystodes, trematodes, and nematodes. So then the general characteristics of what is the general characteristics of cystodes? General characteristics means their morphologically forms, their adult forms, their egg or larval forms. Some of the similarity are there in that species, and then in that one there are different parasites are there. So when you know the exact general characteristics of that category of that parasite, then you can have correlate the other things about that parasite. So this is very much important to let you know, because if you know the particular parasites that cystosomes comes under the trematodes, then you can easily write down the morphology of that cystodes. What is that morphology of that parasite, that it belongs to trematode. That means it's a leaf-like shape and all things. So that therefore you must know the general characteristics of this. So now I am going to revise the general characteristics of this trematode again. So in the general characteristics, you can see here they are also called as the flukes. So that is the first one. Okay, so they call as flukes. So therefore they are called as liver fluke, lung fluke. Depending upon their habitat, they can call as a liver fluke, intestinal flukes, lung flukes, and all. So or the blood flukes. The blood flukes are the cystosomes, which you already learned. So now then their body is unsegmented. As you can see, the cystodes and nematodes, they are segmented, and or the continuous one like this. Here is also unsegmented body, flat and leaf-like. That is the specular characteristic. So they are, when you see the worms, they are not typically worm-like elongated shape. They are having the flat and the leaf-like shape of the adults. Then about their reproductive systems, they are hermaphrodites. That is mean they are monoecious. That means the single adult is having both male and female sexual reproductive organs in the same adult. Okay, so that is important in this as a general. Having exception to this general characteristics, the other what we are going to see today, they all are hermaphrodites. That is the monoecious. So the cystosomes having the male and female parasites different, but here what we are going to learn today is all parasites having the single adult having both sexual characteristics in the same adult. Then again they are oviparous. That means they lay eggs. Okay, so these are egg-laying organisms. Second, and the next is they have the larval form. So when you see a general characteristic of the trematodes like this, they are having an adult form, the egg form, and the larval forms. That you must know. Why? Because depending upon this, you are going to explain the life cycle of these organisms. So that is important. Which forms is present in which host? According to that, you must complete the life cycle. So therefore, this is important. So these are larval forms are also there. Not a single larval form. They have 5 different larval forms. So that is important. Okay, 5 different larvae. That is common for whatever organisms, whatever parasites we are learning. I am going to teach today, that all you having these 5 larval forms in their life cycle. So the larval forms will be Miracidium first, then Sporocyst, then Radia. Radia will be having first generation and second generation radial, and then Cercaria and Metacercaria. Okay, so these are the 5 larval forms which can be seen, observed in this one. Then the life cycle, when we discuss of these trematodes today, they were having one definitive host, that will be either man, domestic and wild animals, and 2 intermediate hosts. That is to be noted. That is, they have 2 intermediate hosts for their development. So intermediate host, and first intermediate host will be freshwater snail or molluscus. So most of the time, these all trematodes, whatever we are going to learn today, they are having the first intermediate as a freshwater snail. Then the second intermediate will be depending upon the which parasite is there. It will change. A majority of them having the second intermediate host as the aquatic plants. Okay, so the aquatic plants will be their second intermediate host, or the freshwater fish or the crabs. These are will be the second intermediate host. So again, these general characteristics you must remember. Whether they are unsegmented, flat, leaf-like, trematodes means that clear definition will be unsegmented, flat and leaf-like adults. Then they are hermaphrodites, lays eggs, and they have 5 different larval forms. And life cycle, 1 definitive and 2 intermediate forms. So these general characteristics you write it down, and again whenever we are going to learn any new parasite in this category, you have to write it down what are the forms and what are the hosts and all this. So you are going to learn easily about the life cycle. Now, what trematodes we are going to see today are already we have learned about the blood fluids, that is cystosomes, and that you have seen where the habitat is there. Cystosoma will be the long question for you in the exams. Okay, so right now, so cystosomes is a long question. So in details, you must know the classification, what are the separate species that are there in that one, about the blood fluids, depending upon the habitat, where they located, and their life cycle and all their morphological forms. Okay, so here we are going to learn today is the liver fluke, then intestinal flukes, and then lung flukes. So in that liver flukes, we are going to see the 2 parasites, that is Fasciola hepatica and Clonorchis sinensis. Okay, and in the intestinal flukes, we are going to learn about the Fasciolopsis buski, and the lung flukes we are going to see about the Paragonimus westermani. Okay, so these are very difficult words too, because it is the first time you are encountered with such words. So write it down and repeatedly revision it, so you can understand these words immediately. Because you may have a short note or like 1 mark questions for that you have to enumerate. So they will make you ask you about the enumerate the 2 liver flukes. Okay, so that you have to must know the Fasciola hepatica and Clonorchis sinensis as the name. Then you got to 1 mark. Okay, so this is for multiple choice question, and short notes they can ask. So for long case, is always asked on the cystosomes. So now we are going to learn each of these parasites in the detail. So the first, Fasciola hepatica. Fasciola hepatica is a common liver fluke. Okay, so this I already told you, this is a liver fluke. Okay, so that means its habitat is again the liver and the bile tract. The disease what is called by this Fasciola hepatica is known as fasciolosis. Okay, fasciolitis. Then it is a zoonotic type of disease, endemic in mainly sheep raising countries, like in the world Peru, Bolivia, and Chile. These are the India. They are the sheep raising mainly countries. So these countries are mainly having infections with such parasites. In India, it's really uncommon, but few cases have been reported from the northeast region of this our country, that is Assam, Uttar Pradesh, Bihar, and 1 and 2 cases have been also reported from the Mumbai and Vellore. But what is the thing is what we have seen in our India, that is the Fasciola hepatica is less common than the Fasciola gigantica. So the Fasciola gigantica is the main liver fluke that has been reported mainly from the India, not that of the Fasciola hepatica. Fasciola hepatica having worldwide predominance, but in India we have reported Fasciola gigantica is the main cause of this liver fluke, causing the liver disease. So in the adult form, so now we are going to see the adult form mainly, and then we are going to directly on the life cycle of this one. The adult form, as you can see, these are flat, leaf-like structures. Okay, not like cystodes, like elongated ones. So here are the flat, leaf-like structure. You can see these are the adults, which is having. This is about the Fasciola hepatica, and this is the Fasciola gigantica. Okay, so these are the size-wise, is a more size bigger than this hepatica. Therefore, it's called the gigantica. Okay, so the Here the size of the fascia hepatica is 3 centimeter in the length. So this is about 3 centimeter in the length and 1.2 centimeter in the breadth, about 1.2 to 1.5 breadth. Okay, that is the size of the fascia hepatica adult. Then they are mainly the brown color, and you can see the anterior end is slightly prominent. You can easily see the prominent end, and they have one of the type of this shoulder. You can say these are the shoulder, like two shoulders. You can clearly observe in the fascia hepatica. In fascia gigantica, again the same, the size is bigger, but the prominence is also there in the anterior end, but the shoulders are not that prominent as you compare to the fascia hepatica. So this is about the adults of the fascia species. Then again, as I told you, they are the hermaphrodite, that is having both of these male and female sexual characteristics. Along with that, they have the suckers. Okay, so they have the suckers also. They have the one is the anterior end. They have the folical projection here, and they contain the oral suckers. Okay, 1 minute. Huh? Okay, so these are the anterior end having the projection, which is having a mouth, and this is the oral sucker, and there is a the ventral sucker is also situated, which is away from this oral sucker. Here, your acetabulum, what you know now, this is the ventral sucker. So they, with the help of these two suckers, they are going to attach to the biliary duct and the newer tissue parenchyma with this the help of these two suckers. And then this is the gastrointestinal tract of this fascia hepatica. While these are the diagram of this the reproductive system. So here you can see the female genital aperture, female genital organ, then the male genital, like male genital aperture, then ejaculated duct, and then all ovary, oviduct. Here all vast difference of the male genital tract. Then these are all is present in the single adult womb. Therefore, they are the hermaphrodites, and which lays eggs. Now they lays eggs which are unembryonated. Okay, so now we are going to see their life cycle in the detail. So life cycle, when we see, we'll start with the first definitive host, intermediate host, and second and third one is the route of transmission. So here in fascia hepatica, the definitive host is predominantly the sheep. Okay, the sheep is the definitive host. Then also some of the goats, cattle, and even man can be a definitive host. And the first intermediate host, as I already told you, will be the snails. So snails are the first intermediate host, and the second intermediate host is these aquatic plants. So the definitive host will be the man, sheep, cattle, okay, goats, and the intermediate host, the first intermediate host is the snail. The second intermediate host will be the aquatic plants. So, and the route of transmission is definitely the aquatic plants which were been eaten or which were been ingested by the human or any of the cattle or sheep. Okay, so there will be a infection from the of this fascia hepatica, because this aquatic plant is harboring the encysted forms of the metacercaria. Okay, metacercaria is the larval form of the this parasite. The 5 larval forms which we are going to see in the detail now. So the last larval form is the metacercaria, which is encysted like a cyst form, and which are present on these aquatic plants. And when these aquatic plants is eaten by or ingested by the cattle, sheep, or the man, we do not proper washing or not proper cooking, the man can have this aquatic plants, and the sheep and the cattle they do they ingest it enter the man on sheep and all things. So what will happen when they ingested into the intestine? They go up to the duodenum. Then the encysted forms of this metacercaria, this metacercaria will encysted exist. They come out of the cyst, and then they from the intestinal tract, they penetrate the intestinal tract and reach up to the peritoneal cavity. Okay, so the peritoneal cavity will reach there, and then they travel up to the upwards to the liver. They directly penetrate the liver parenchyma and reach into the hepatic zone, hepatic parenchyma, and where they reside into the hepatic ducts. and then hepatic ducts, they were turned into the adult form. Okay, so now in the hepatic ducts and in liver parenchyma, they will reside till reach up the metacardia will reach there, and then again it turn out they change their morphology and then it turns into the adult form. Okay, so what will happen? It takes about 9 weeks. Okay, it takes about 9 weeks of infection to achieve this adult form. So after injection of such metacardia reaching up to the hepatic parenchyma, residing there, and again changing their shape or the morphology to the adult form, it will take around 9 weeks. Then whenever it is present in this one, they start laying these eggs. Okay, so they start laying these unembryonated eggs. Okay, they start laying unembryonated eggs, which travel to the hepatic duct, bile duct, into the intestinal duodenum, in the duodenum, and through the duodenum they get intestinal tract via the tract. They will excrete into the stool like an embryo and unembryonated egg. So the unembryonated eggs will be excreted. So this is the phase where we can detect it. So after such infection, we can have a stool examination to see the unembryonated eggs with the passes in the stools that will be there. So these unembryonated eggs which are in pass, they go into the water, and then next stage of their development will start in this water. So the eggs develop in the water in 1 to 2 weeks to release the mycelium. Okay, so the next form of this larval form is the mycelia. Okay, so this unembryonated goes in the water, takes 1 to 2 weeks for development. Eggs becomes unembryonated in water, and then they transform into the mycelia. This seed, it is a which is hatched from the eggs and stick out. The mycelia comes out this larval form, and then it will travel into the water in search of the first intermediate host, that is a freshwater snail. So now this mycelia when reach up to the snail, they penetrate the small intestine of the host. They go inside the by the water. They go inside the intestinal tract of the snail. They penetrate the small intestine of the snail, and then they transforms into the remaining forms of their larval form, that is the sporocyst, the radia, first, second generation, and ultimately cercaria. Okay, so now when the cercaria has been developed in the first intermediate host, that is snail, these cercaria again existed and released from the snail into again water. Then the free cercaria is now present in the water. They now in the search of the aquatic plants. So the cercaria, when they can have a this is a tail like this thing. So they can travel, they can move around in this one and reach up to these aquatic plants. When they reach up to the aquatic plant, in their leaf on their stem, they reside there, and then they form cyst. So they encircle themselves and they form cyst. So then this cyst has been formed, and in that cyst, the metacercaria, the last stage of the larva, which is infective form also to the definitive host, that will be turned out to be here. And then again, this has been raw eating of this plant will lead to the infection. Okay, so this is the life cycle of the Fasciola hepatica. Similarly, life cycle of the other species also there, but minor changes are there. Therefore, when you can merge or you can understand this first life cycle, it will be very easy. Okay, so the first thing, when they reach up to the liver by the penetrating penetrative tract, they transform into the adult worm, lays the unembryonated eggs in the water, which again start developing like embryonated eggs, and then their mycelia. The mycelia will hatch out of the egg and comes out. Again, we have small flagella-like things, so they can travel up to the snail, first intermediate host. In the snail, they will again transform into the remaining forms of the larval forms, that is the sporocyst, first, second generation radia, and ultimately the cercaria. The cercaria forms, when it is formed, it gets released into the water from the snail. It has a tail. From that tail, it can travel and reach up to the second intermediate host, that is the plant, aquatic plants, and where they again encysted themselves to form a encysted forms of the metacercaria, which will be ingested, and again the life cycle will be completed. Okay, so this is the life cycle of the Fasciola hepatica. Now the pathogenesis. The pathogenesis, as you know, when we ingest such encysted metacercaria, they will hatch out and come and they penetrate from the intestinal tract. They come into the peritoneal cavity and they migrate up to the liver. So during that phase, and they migrate up to the liver, encroaches the parenchymal system of the liver, and they then when they reach the liver tissue, then they change their shape into the adult form. So during this migration and happening of things, the patient may have a fever episode and right abdominal pain, right upper quadrant abdominal pain. Okay, so that will be the acute phase. The first 8 weeks or 9 weeks, the patient may have intermittent fever, intermittent right upper quadrant abdominal pain like things. Ultimately, when this reach and it goes into the higher form, then it takes more time. Then the hepatomegaly also can be on palpation you can find the hepatomegaly. And on the blood examination, definitely any parasitic infection, you will have raised counts, total counts. Along with that, eosinophilic count will be more. So that is the diagnostic one. When adult worm is formed, they will reside into the liver parenchyma as well as hepatic duct. It travels also down to the bile duct. So ultimately, they can obstruct. The diameter, the length of this adult is about 3 centimeters. Okay, so about 3 centimeter diameter, the 3 centimeter length, and 1.5 centimeter of the breadth. So that when it reaches to the bile duct, it obviously will block. Okay, so there will be obstruction of the bile duct. Then, if the chronic phase, that means it will remain for last 6 months, then the remaining the adult form will form adult worm will form a multiple subcapsular liver abscesses. Okay, and also it may lead to biliary cirrhosis, but in less common causes of biliary cirrhosis, but it may lead in chronic cases may lead to chronic biliary cirrhosis. So these are the pathogenesis of the facial hepatica. For lab diagnosis, as I already told you, the stool examination after 8 weeks, when the adult form is generated, and adult form start laying eggs after 1 to 2 weeks. So near about after ingestion of such infected metacercaria, he will be passing the eggs in the stool. It will take around 10 to 12 months to pass this one. Okay, the 3 to 4 weeks, at least 3 months. After that, he will do the stool examination. So you can see such of the oval, bile-stained, operculated eggs in the stool examination. But there will be a count may be less. So whenever such history is there of the patient, you must do the stool. Before doing the direct stool microscopy, you have to do the concentration technique. Okay, so here we are using sedimentation technique of the stool concentration, not the flotation technique, because these are some of heavy eggs, so they get settled, sedimented at the bottom. So therefore, we are using the sedimentation technique for stool concentration method, and then we'll do stool microscopy when you can see oval, bile-stained, operculated eggs can be seen. Again, antibody detection you can do by ELISA or counter immunoelectrophoresis, or also by Western blot technique you can do antibody detection. And now the PCR methods also available to detect facial hepatica. The specific gene from this stool specimen can be used for the diagnosis. Okay, so this is about the first liver fluke, that is the facial hepatica. Now we are going to do the second one. Liver fluke is the Clonorchis sinensis. Okay, so Clonorchis sinensis is also known as the Chinese or Oriental liver fluke. Okay, because these are mainly found in the Eastern Asia like China, Korea, Japan, and Malaysia. Okay. Here the adult length is somewhat less than that of the facial hepatica. Here the length is only 1 to 2 centimeter, while the breadth is still smaller, that's only 3 to 5 millimeter. That is only half centimeter up to the breadth is there. Okay, and they are more likely the elongated one. Okay, elongated flat one. These adult worms produce near about 1,000 to 4,000 eggs per day. So therefore, the load of this organism, the parasite, will be higher in such cases, leading to even cholangiocarcinoma, bile duct carcinoma. Okay. So again here we can see the Clonorchis sinensis. Here the definitive host is the man or dog, pig, cat, and the rat. So and it's the animal and domestic as well as the wild animals. And the man is definitely there. The dog, cat, pigs, rats, and all can be a definitive host. Second, the intermediate host. First intermediate host again the snail. snails are the first intermediate host, but only thing is the second intermediate host is the here freshwater fish. Okay, not the aquatic plants. Here the freshwater fish are the second intermediate host. So again, mode of transmission will be encysted metacercaria. Okay, the metacercaria which is present the metacercaria in the flesh of the skin. Here they are not encysted form. They are directly present inside the flesh or the skin of this fish. So whenever undercooked fish, eating of the undercooked fish or not properly cooked fish, so that may lead into this metacercaria to reach up to the duodenum. Again, here exists in the duodenum, then metacercaria will be released. Okay, then this metacercaria again from the peritoneal cavity reach up to the liver and penetrates the liver tissue, parenchyma, and then it resides there and develop into the adult. So this adult will develop into liver. Again, they travel up to the biliary tract. So biliary tract is mainly the site or the habitat of this Clonorchis sinensis. So in the biliary tract, they reside there. The adult will form. This adult will form now start laying the eggs, around 1 to 4,000 eggs per day. Only thing is the eggs are the embryonated eggs. So whatever we have seen previously of the facial hepatica, that is unembryonated eggs which are laid, and that turns to embryonated eggs into the water. But here already embryonated eggs has been laid, released into the intestinal tract, which has been excreted along with the feces into the water. So here embryonated eggs pass in the feces, which comes again into the water. Here again what we have seen last time, the miracilia which has been released into the water, and then it goes into the snail. But here what will happen? These eggs will go inside the ingested. The small eggs will get ingested by the snail like the first intermediate host, and here the all rest of these larval forms will be developed like miracilia, sporosis, Redia form, and the Cercaria form. Previously in sexual hepatica, this miracilium form is not formed in the snail, which is formed in the fresh water, and then this miracilia will infect the snail. Here in Clonorchis sinensis, this egg is directly ingested by the snail, and then all of these four larval forms, that miracilia, sporosis, Redia, and Cercaria, will be developed into the host, that is a first intermediate host like a snail. And then this Cercaria, which is ultimate fourth form of this larval form of this parasite, which has been released again from the snail, and then it travels and gets ingested free swimming. They can swim around and reach up to the fish. Then when fish, what will happen? The fish get ingest the Cercaria, and then will reach up to their muscles and the skin of the fish, and then here they get encysted. Okay, so the metacercaria, they encysted, and then the forms, the Cercaria will turn into the metacercarial forms. Encysted metacercarial forms will be formed here in the muscles and the skin of this one, which get ingested, and again the life cycle will be generated. So here the only difference between facial hepatica and then Clonorchis sinensis is the all the larval forms will be in the snail, and the whatever eggs has been laid by the human or the man will be the embryonated egg, not the unembryonated, like in facial hepatica. So pathogenesis, depending on the burden of this parasite, if the worms are very less in the number of light worms are burden, then it will be asymptomatic. Then the chronic or the heavy burden, when the increases the number of the adult will increases, then there will become mechanical obstruction of the bile duct. And again, here is the most common cause of the cholangiocarcinoma. That means the bile duct carcinoma is common with this type of the infection. That is okay. So Clonorchis sinensis is one of the cause of the bile duct carcinoma. But along with that, not only the infection, other risk factors like old age and all other risk factors will be there to be induced into the cholangiocarcinoma. Okay. So this is about the pathogenesis of this Clonorchis sinensis. Now in the stool examination, laboratory diagnosis, you do the stool examination. Here the only difference is these are the flask shape. Okay, these are the flask shape of that this egg, embryonated flask shaped egg, and shows small knob. Here you can see a projection of the knob at the posterior end of the opercular end. Okay, so this is the diagnosis of the characteristics of the diagnosis of Clonorchis sinensis. Even you can do the stool examination is also helpful, but this you can directly aspirate duodenal aspirate can be done, and this aspirate can be examined under microscope for similar eggs to be seen in the microscopy. So duodenal aspirate will be the more having more sensitivity of such for the microscopy, which can be done like entero test for the duodenal sampling. The entero test is also done for the giardiasis. Okay, so like similarly, you have to do the entero test and take a sample from the duodenal area, and then the microscopy has been done, and where we can see such egg forms of this clonarchis sinensis. Then antigen antibody detection by ELISA and PCR methods also now available for diagnosis from the stool or the directly from the duodenal aspirate. Okay, so this is about the clonarchis sinensis. Now we are going to move on the intestinal fluids. The intestinal fluids are the fasciolopsis busci. So these are we are going to see fasciolopsis busci. Fasciolopsis busci is the giant intestinal fluid. Okay, so that means habitat is the small intestine. Will be the of this parasite. These are also found in the duodenum and jejunum, as I already told you, and endemic in again Southeast Asian countries like China, India, Pakistan, Bangladesh, Thailand, Malaysia. India prominently Uttar Pradesh, Bihar, Assam, West Bengal, and even Maharashtra has reported some of the cases. But mostly Uttar Pradesh, Bihar, Assam, West Bengal. These are the majority of the cases that are reported from this area. The risk factor has been seen that the poverty, more poverty, unhygienic socio-cultural practices, food habits, that will all lead into this intestinal fasciolopsis busci infection. Okay. Now the adult of the fasciolopsis busci is around 2 to 7.5 centimeters in length. So this is the longest worm as compared to that hepatic fluids. Okay, so the hepatic fluids are small, 3 centimeters wide, but here it is about the 7 centimeter of the length, and breadth is only about 1 to 2 centimeters, and thickness is about 0.5 to 3 millimeter in thickness. Again, they are flexi-like things, and with the broad anterior end. Here anterior end is not that much pointed, as we are saying projection pointed are there in the case of the fasciola hepatica cases, but here we can see the anterior end is not that of broad, and it also does not have the cephalic cone, which is present. Then like the cephalic cone, that means the shoulder-like structure. Okay, they don't have any shoulder-like structure. The typical leaf-like structure can be seen of this fasciolopsis busci. Okay. It has two structures, again the same, that oral one and the ventral one, and they are also the hermaphrodite one. So here the oral structure and here the ventral structure, which is acetabulum, and they are also hermaphrodite, that is having the testes and ovaries, that is both male and female reproductive organs in the same species, same anatomy. Here the adult, the life cycle when we see of this one, this is the definitive host is the man and the pig, while the intermediate host again here the snails, and second intermediate host is again here the plants like Fasciola hepatica. This one is also having the second intermediate as a plant. So here again the same thing here happening. Metacarcaria, which are ancestor forms on the plant, which get ingested by the pig or the definitive host man, pig or man. Then it reaches up to the duodenum. Here they exist in the duodenum and itself resides in the small intestine. They do not travel because it's an intestinal fluid, so they reside in the intestinal duodenum and jejunal part, small intestine, and they turn out into the adult form. And in adult form, according to their oral structure and ventral structure, they get attached to the intestinal wall, that duodenal and jejunal wall, and then they develop their into the total adult form, and then start laying the unembryonated eggs. Now the unembryonated eggs get extruded into the water. Again, this turns into the embryonated egg. Again, in the miracidia, the first larval form. Now, this miracidia will be ingested by the snail. Then snail get in porosis, radia, cercarial forms, and then cercaria will travel into the water, reach up to the aquatic plants to get encysted itself and again form the infective form. So, this is all similar to that of the Fasciola hepatica. but here only they will not reach up to the liver because their habitat is the intestinal tract. So they will repair into the intestinal tract. Okay, so then its pathogenesis, the light infection, that means when the load is less, then there is no infection. There is no symptoms like asymptomatic one. But when it's severe infection, it will be reported partial obstruction of the intestinal tract because these are the 7 centimeter, about the 5 to 7 centimeter length vein. So whenever the load is more, the intestinal wall, the intestinal lumen get occluded. Okay, because of this high load of this parasite in the intestinal wall. Okay, therefore the occlusion will be found, partial obstruction like symptoms will be there in severe infections. And again, these are the 7 centimeter long, so they will reside they are attaching to the intestinal wall and covering the all intestine. So therefore, the absorption will be less. Absorption of vital nutrients will be hampered in such cases. So there will be a malabsorption of the patient, or protein losing enteropathy will be the cause, will be the next symptoms, and then the protein losing enteropathy will be showing a yellowish green type of stool. The patient may show yellowish green type because protein loss. Protein will be not get absorbed into the small intestine. Therefore, the protein gets excreted into the stool, which is resulting in the yellowish green stool along with the malabsorption. So, this will be the pathogenesis or the symptoms of the patient in the severe infection. So, lab diagnosis, again stool examination will be done for unembryonated eggs, and here again it will not any such parasite, rare parasite, we have to do the sedimentation or stool concentration technique, and when we suspect of trematodes, then we have to do for the sedimentation methods of the stool concentration. So here you can see the operculated eggs in the stools. One minute. Okay, next one. The lab diagnosis of this one, facial opsis bursky, that is intestinal liver, intestinal fluids. Here, a stool concentration method done by sedimentation method, and in the microscopy you can see operculated stool, operculated eggs in the stool. But the definitive diagnosis, as I told you, there may be a what is symptomatically you can't differentiate between the intestinal one or the liver fluke are there. So for the diagnosis, you can also go for the radio diagnosis like sonography, CT scans. Therefore, you can see a cyst forms can be formed into the liver, or maybe a load of this parasitic load can be observed whenever in the X-rays or in the CT scans and sonography. So definitive diagnosis by the identification of the adult one. So when you can take that and if you're operating for the obstruction, you can find out the directly exposed to that parasite, so that parasite can be sent for the identification for the lab, whether it is the facial hepatica or whether it is a facial opsis bursky. Okay, so that will be confirmed by the adult examination. Now we move on to the we have to just enumerate the other intestinal trematodes. Okay, so as I told you, there may be ask you the question about the enumerate the 2 or 3 names of the intestinal trematodes. So in that you can write down that facial opsis bursky is the one, then gastrodiscoidus hominis, then heterophiles, then Watsoni, and the 4th one is the Metagonimus yokogawi. That is the 4 other intestinal trematodes which can cause infection to the man. But these are very rare. But you must know at least 2 to 3 names of the intestinal fluids. Now we are going to see the last one is the lung fluke. That is the Paragonimus westermanni. Okay, Paragonimus westermanni is also known as the oriental lung fluke because its habitat is the respiratory tract. That is the lungs where they are going to reside. it causes endemic hemoptysis in man. Okay, so they were reported some of the in area there will be all patients are majority of the cases are reported presenting as a hemoptysis, and then they found to be this is the paragonymous Westermanni infection happening to the particular area, resulting in the raise in this increase in such cases of hemoptysis cases. So North America and Europe is the endemic in the are the area where this infection is very endemic, and in India northeast state like Manipur has reported paragonymous Westermanni infections. The adult ones lives in the parenchyma of the lung, which is the habitat of this particular organism. The adult worm is again a thick, fleshy, reddish brown, flat, leaf-like. It is having 16 millimeter in the length, like 1.5 centimeter in the length, to 8 millimeter in the breadth. Okay, so the length and breadth. Here you can see oval in shape, but the anterior end is broader. Okay, so what we are intestinal fluid, liver fluid mean anterior is normally pointed or something like that compared to that one posterior one. Here the anterior one is the somewhat broader as compared to the posterior one. And again it having a oral sucker situated anteriorly and a ventral sucker situated in the middle of the body for the attachment of this adult. Again the life cycle which we see, they have definitive hosts like man, dog and cat, and the intermediate host, again the first intermediate host is the snail, while the second intermediate host here is the crab. Okay, the crab or crayfish. These are the second intermediate host. So if you see these all organisms parasites, the first intermediate host is the snail only. Only you have to remember the second intermediate host for the particular parasite. Okay, so here the second intermediate host is the crabs. So again the same thing, Cercaria invades the crustaceans or the crabs and then ensists into the metacercaria. So the crab which is having metacercal encysted forms of metacercaria in the flesh of that crab will be the infective form. And when ingestion transmission from it, not raw eating of this crab or improper cooking or undercooked crab eating, that may lead into the cercaria will reach up to the intestine. Okay, so what happens in the intestine? So from the intestine so from the larva which has been reached up to the small intestine, that larva, that metacercaria, which escapes out, that exists in the small intestine, and then penetrate the intestinal wall and reach up to the peritoneal cavity, as we have seen with the fasciola hepatica. But fasciola hepatica travel up to the liver, but what is its residing site or habitat is the lung parenchyma. So then they pierce the diaphragm and they reach up to the lung parenchyma, and then they settle near the bronchus. Okay, so they travel the metacercaria from the peritoneal cavity, they travel through the diaphragm, and then it reach up to the lung parenchyma to reach up to the bronchioles of this lung parenchyma. So they will reside there, and then they mature there into the adult form. Okay, and then when they mature into the adult form, they start laying the unembryonated eggs. Okay, so they start laying the unembryonated eggs and they at the size of the lungs. So here they reach up to the bronchial lung parenchyma, they reside there, and then they turn into the adult form, and then they start laying the unembryonated eggs. Why they reside near the bronchiole? Because the unembryonated eggs which have been born into the bronchiole, they shed or they release their eggs into the bronchioles. So the bronchioles, whatever mucus is there, along with that, it gets cupped out. Okay, therefore it gets cupped out. So therefore, what will happen? They get cut out along with the sputum. So in the sputum, they are having unembryonated eggs. Along with that, when they cupped out, some of the sputum get swallowed. Not all the sputum get expectorated, but some of the sputum get swallowed. Along with that, the unembryonated egg will again go into the intestine. And in the intestine, they will not turn into any change, but they, as it is, they get excreted through the feces. So the unembryonated eggs can be on the diagnostic phase of this parasite, where can be found in the sputum and as well as into the feces, but mainly the sputum. Okay, so then unembryonated egg it turns into the embryonated eggs into the water. The miracidia which will hatch out from this, and then they travel up to the snail. In the snail, then the rest of this larval form, there is porosis, lydia, cercaria. Cercaria will get released. That released cercaria which penetrate into the ingested by this crab or crayfish, and then get insisted into the metacercarial forms, and when it becomes the infective form of this one. Okay, so this is the complete life cycle. Here, what is happening irrespective of the liver, they reach perforate the diaphragm and reach up to the lung parenchyma, reside near the bronchioles, where they get expectorated. Okay, they get hemoptysis, or because of this, they are traveling, so there will be a minor bleeding will be there. Okay, so whenever this unembryonated egg get laid down into the bronchioles, along with that, some of the blood also be there, and therefore, whenever patient comes out, then along with that, the blood thing in the sputum will come. That is known as the hemoptysis. Okay, so the hemoptysis will be the symptom of this patient. Now, pathogenesis, there will be a pulmonary paragonimiasis and extrapulmonary paragonimiasis. Okay, so the pulmonary one, that means when the adult worm initially parenchyma, what are the symptoms in that one? So, the pathogenesis, what will happen? They form eosinophilic granulomatous inflammation. Okay, so these are the terms which you going to see into the already you have learned in the pathology. There will be a granulomatous inflammation, eosinophilic mainly because it is a parasitic one. So there will be a granulomatous inflammation in the lung, which leads to formation of the fibrotic capsule around the worm. Okay, the encapsulating a fibrotic capsule or a cyst will be formed around this worm. So these are commonly seen in the right lung. We don't know why it is so, because it's intestinal travel through the diaphragm and all things, but it is mostly seen in the right lung. These cystic forms of the fibrotic capsules, which are enclosing the adult worm. And then cysts containing the blood mixed the thick purulent fluid, because the adult worm laying eggs there, and also they are encroaching this along with that there will be damages to the microvascular blood vessels. Then leads to the containing the blood mixed the thick purulent fluid will be there into the cyst and along with the fluids and eggs. So then when the cyst breaks up into the bronchioles, so this all blood mixed the sputum will get extracted. This is the pathogenesis of the pulmonary paragonimiasis. Extrapulmonary one, what will happen? The worm will reside in the lungs, but sometimes they migrate from the ruptured cyst up to the various sites, like liver or spleen or any abdominal wall. So there will be again there cyst formation in that particular area. There is the extrapulmonary paragonimiasis, but these are rarely seen. Lagonosis is again done from the unembryonated egg are seen under the microscopy, which can be seen by sputum microscopy, right? So the early morning deeply cuffed sputum are the sample first to be collected in such patients. Okay, so the early morning deeply cuffed sputum sample, they have been done in the routine saline mount. They can see such eggs can be seen, unembryonated eggs populated, and where the bird and also some of the tissue can be taken, a lung biopsy can be taken, which can be stained with this HID staining. You can see such cystic forms, you can see the cystic fibrous forms, and inside that there is a adult of this parasite can be seen. Okay, so this Paragonimus westermani adult can be seen in this cyst form of the lung biopsy tissue. And also, this is the sputum of the collected patient for examined under the microscopy. So the multiple sputum examinations should be done. That should be done at least 5 to 10 specimens. Only one sputum examination which has stain and comes negative, that will not give you particular idea. So you need to stain at least 5 to 10 such early morning sputum samples for every day to see the positivity, to come the positivity. And again, when it reaches to the lab, you have to do the formalin ether sedimentation technique, so that whenever scanty 2-3 eggs will be there, that gets sedimented easily, and the positivity rate will be more in such cases. Okay, so this is all about this today's topic, that is the Trematodes II, and this that means apart from this topic, we have learned other trematodes that are liver fluke, lung fluke, and intestinal fluke. So parasites and the life cycle. If you know life cycle of one cycle, you can easily make out the life cycle of the other ones. Okay. So you must know about these things. Okay. So this is all about it today's lecture. So next lecture will be when and all this has to be communicated in short period of time. Okay. Thank you.